This is a brief tutorial on ways that we can organize a scientific manuscript to be clear for our readers and to make sure that our methodology and our results match, and that our discussion points are clear to our results, and that our introduction clearly introduces the study that the reader is about to read. For this tutorial, let's think about a manuscript as an hourglass. And let's start with the top half of the hourglass. Our first job is to introduce our reader to the topic. So we're going to provide our introduction, and sometimes we'll also see this referred to as the background. And in this case, we use the widest part of the triangle to illustrate that we're going to give a broad introduction to the topic. In this example, we're going to talk about a study that I co-authored looking at the potential distribution of anthrax in Kyrgyzstan. So we first have to introduce our reader to Kyrgyzstan and possibly this larger region of Central Asia, and the fact that anthrax is a zoonotic disease of great concern in this region because of the agrarian lifestyle of many rural farmers in Kyrgyzstan and limited veterinary and human health care in the region, which could increase the risk of human cases that were unattended to. So our introduction would cover topics such as anthrax as a disease, such as its role as a zoonotic disease, its risk, and possibly risk in the region. So we're narrowing down from the widest introduction to more specific. In the case of our study in Kyrgyzstan, we want to predict where that risk might be. Predict spatial risk. And in this particular study, we do that with something called an ecological niche model. So I'd need to tell my readers something about ecological niche models and what that is as a tool to predict a potential distribution of a species, in this case the bacteria Bacillus anthracis that causes anthrax, using environmental covariates or raster data sets that describe the environmental conditions. At this point, our reader has now some information on the disease that we're interested in, the risk that it poses, and the region that we're performing our study in, and the kinds of tools we're going to use broadly, like some introductory material about what ecological niche models are and how they're used. And then, as we narrow down to the end of our introduction here, we get to the specific objectives of the study that we're going to present. So the specific objectives we would give our reader, for example, in this study, our goal was to use historical anthrax outbreak locations and ecological niche modeling to better estimate the potential distribution of anthrax in Kyrgyzstan to inform public health of risk areas. So now we've framed our background, our introduction, and our study objectives. So the reader now knows what the broader context of our study is, predicting anthrax or disease risk, specific tools to do that, ecological niche models, and specific objectives, using those tools and data from Kyrgyzstan to predict the risk in a specific place. Now, let's put together the rest of our hourglass. Now our job is to start putting together our methods. And our job here is to describe for our reader how we achieve those specific objectives with specific methods. And this is where we're going to describe our data sources, our data sets, and our analysis that's performed so that the results can be reviewed. In this study, we have our historical anthrax outbreaks, and then those are divided into two parts, model training and model testing data. That's the way that our data sets need to be divided so that we can perform that particular type of analysis. So here in the methodology, I would have a figure that shows the map here on screen, and I've subdivided the data into two parts. That would be described in the methodology, and then the reader would be aware of the sources of data that I had. It would be described where these data came from and how that division was made. So here we would describe our data source, or sources. So we had outbreak locations, and we had covariates describing the environment. Those are the two types of data needed for an ecological niche model. So this section would provide detail on the source of those outbreaks. The Kyrgyz public health system is described, and the source of the covariates, some global sources of data that were downloaded. And those data would be described visually with the map I showed a moment ago, and may also be described with a series of tables and references. In this particular study, I used an ecological niche modeling package referred to as GARP, the Genetic Algorithm for Rule Set Prediction. So this is where I would have a brief overview and references to that software, a description of how the model works, and some information about the parameter settings. So how did I use that modeling system in this particular study? One thing that we want to note about this method section is that this is the section of the paper we provide a true recipe for our reader. And a recipe is meant to be repeatable. So it's in this section that we need to be clear in the data that we had, the methodology that we employed, so that we can interpret those results correctly. And in order to be scientific, we have to be able to produce methods that can be repeated by another analyst. Our next section here in our hourglass is our results section. And this is going to be your record of outputs. Just like our methodology section was a recipe for how to prepare those results, our results are a direct accounting of what we found in the study. Not a discussion or a direct interpretation of that. That'll come later in the discussion. In the results section, we're going to present our results. In this study, our objective was to produce an ecological niche model-based prediction of where we anticipate Bacillus anthracis, the pathogen that causes anthrax, is most likely to persist in the environment. And so our results, our primary results for this study, were the production of this map shown here. As I mentioned earlier, this was a GARP study, so we can see here that we're getting a GARP output. We have a map of Kyrgyzstan, and we have some indication of where the likelihood is highest. That's indicated in the red areas. So very quickly, our reader can draw their attention to these areas and see the greatest areas of risk for the likelihood of the anthrax pathogen to be found. In the results section, it's important that we don't over-discuss these results, but rather just report them to the reader. Now, in the case of a study where a map is one of our primary outputs, we want the results section, the narrative of the results section, to have a clear description of the map. So if we were to just bullet what we might say about this map, we might identify that the areas around Karakol, Bishkek, Talas, and Osh were the areas of highest predicted likelihood. So we could say broadly 4 areas support pathogen persistence. And then we would name those areas. Once we've reported our results and our reader has a clear understanding of what we found, it's time to discuss those results. And what we've been doing throughout this section of our paper is going from narrow to broad. Our introduction started broadly and focused down to the specifics of the study, and now we've gone from the specific methodology and results out to our discussion. So our first objective in the discussion is to summarize our study. Often this is done with a brief sentence or a couple of sentences saying, in this study, our objective was to use historical outbreak data and covariates to predict the spatial distribution of anthrax. And a brief sentence or two about our findings. Our next order of business is to now relate that or put that into the context of other studies. So in the introduction, we introduced references, literature, and background on our topic and on our tools, and now we're going to introduce references and literature to put our study into the context of other studies. So it's in this section that we might compare those environmental covariates of Kyrgyzstan to a similar study that was performed in the United States or in neighboring Kazakhstan. So for other studies, we'll look at other like studies, or we might contrast our findings to other methodologies. Okay, so our goal here is to put our work into the context of other studies. In the case of anthrax, previous studies have defined the distribution in Kazakhstan, the neighbor to the north. And so we might compare how the distribution in Kyrgyzstan adjoins or is disjunct from the Kazakh distribution. That would happen in the discussion. Our next goal in the discussion is to present limitations. Every study has limitations. There are limitations on our data source. We never have enough data to make a perfect model. Our modeling system or our analysis of choice will have assumptions that will be described in the methodology and limitations that will be described in the discussion. The GARP modeling system isn't an ideal niche modeling system, so we have to work within the limitations of that. To give a specific example of a limitation, the GARP modeling system is a presence-absence modeling system. So our values are zeros or ones. The pathogen could be present or absent in that cell with some confidence about how well we predicted that. So if we were interested in intensity of anthrax, or where is there more anthrax happening in livestock or humans, that's a limitation that we cannot address in this study. So our methodology is limited to presence-absence, so we cannot make statements. We cannot state intensity. We might also have limitations that our covariates weren't the ideal covariate set for modeling anthrax. So we might make a limitation statement about what we might find if different covariates were available. What's important to keep in mind about limitations is that limitations are a reality and not always or not necessarily a deal breaker. So limitation sections should be framed. We had limitations, however. There are limitations to this study. However, we still provide a first estimate of the geographic distribution of anthrax risk in Kyrgyzstan. So we had limitations. We provide caveats or caution to our reader about those limitations so that we can interpret those results cautiously. Yet we still learn something about the distribution of the disease, and that's something we didn't know before the study started. And then in many studies, though often optional, we might learn something about next steps. What might a next study do to improve upon what's been done in this study? Maybe a better set of covariates, maybe a larger anthrax data set, maybe a modeling approach alternative to GARP. Those are all things that might be prefaced in a paragraph. And then, not all journals include a conclusion section, but often you'll want a summarizing paragraph. In the case of a public health study, that summarization might say, these results can now be used to inform anthrax surveillance or anthrax control measures. In the case of anthrax prevention in humans is usually driven by anthrax control in livestock. So concluding statement might say, these models can be used by veterinary public health officials to identify livestock populations at greatest risk for anthrax by looking at where cattle overlap with the anthrax zones in the figure. And that might be a concluding statement about some future disease control that might come from this new information in this paper. So there we have a brief overview on how to organize a scientific manuscript with a specific example using an ecological niche modeling study and some disease data. Start broadly with an introduction. Let your reader know the value of the study and the context of the study. Narrow down to specific objectives, specific methodologies employed, a discrete and direct accounting or reporting of your results, and then a discussion. One note about the discussion is that the discussion is where, with literature, we can speculate. We want to speculate in an educated approach, but we want to be able to talk about our work in the context of other works. We want to make sure there's a limitation section. We want to caveat that limitation by still reminding the reader that within the context of those limits, we learn something, and then summarize or conclude your study and possibly set up a future study or a future paper. Good luck in your writing, and I hope this helps you get organized.